Well, let's go ahead and get started this morning. We're very fortunate to have Dr. Licht here from the University of Florida. So Dr. Licht actually did his medical training at Columbia University and his residency at the BI, then went across the street or across town, I guess, depending upon how you look at it, over to Dana-Farber for his fellowship, and then did his postdoctoral fellowship also at the Farber in, I didn't know that you could actually get this specialized, eukaryotic transcription. So clearly, again, has had an extraordinary amount of training in this area. He's now director of the University of Florida Cancer Center, but previous to that was chief of Hem/Onc at Northwestern. And a lot of his research really has focused on histone methyltransferases and their impact on not just myeloma, but also acute leukemia and other hematologic disorders. Because of his training and experience in a number of different areas, actually has been serving on the exec committee for ASH, which means if you have questions about dues, go to Jonathan, and is also co-director of the ASH EHA Translational Research Training Program, which we heard a little bit about last night, and is really a neat way of trying to bring hematologic organizations from across the ocean together and train people in unique ways, some of which we might hear a little bit about today. So thank you, Jonathan, for coming up, and we look forward to your talk.
Thank you so much, Sager. Thank you very much, Sager. Thanks very much, Wally, and thanks for you being here this morning. So I'm a medical oncologist who's been working in gene regulation for over 25 years, and we're very interested in how gene regulation dysfunction may drive, in particular we're focused today on blood malignancies and how that understanding may lead in the future to some new therapies. First, I'd like to do some basic review about the basic problem of getting a gene transcribed from DNA into RNA. You need basically some landmarks for this to occur. And what you need is basically our DNA sequences are marked with various specific DNA motifs that recognize various transcription factors, such as the TATA box, which binds a very basic transcription factor, these upstream elements, enhancers that are now known to be very important for regulating gene expression. These are located tens and even 100 kilobases away from where the start site of a gene is transcribed from DNA into RNA. But these are all landmark sites that are then bound by a variety of DNA binding transcription factors that recognize those specific sequences and then just lay various flags down on the DNA. They don't do that much by themselves, but then bring in a series of multi-protein complexes, actually 1 million Daltons of proteins of various complexes, including the RNA polymerase itself, which is going to transcribe DNA into RNA. Additionally, there's a basic problem with gene regulation in that DNA is wrapped around the histone octamer, and this basically inhibits DNA polymerase in anything from happening. Those histones have to be moved out of the way and chemically modified for this transcription to occur. So a number of proteins are brought to acetylate histones, also to methylate them on various regions. And then there's this ATP utilizing remodeling complex, which actually shoves things out of the way to allow the DNA polymerase to go in, open the DNA, melt the DNA, and allow RNA to be transcribed. So there's a lot of things going on at this time. And one of the important things that happens, as I alluded to, is that the histone octamer, seen here in the schematic, it's a basic element of chromatin, the DNA protein complex in our nucleus. 1.5 turns of DNA are wrapped around this, about 145 base pairs. And this basically stabilizes chemically the DNA on the protein, it being positively charged and DNA being negatively charged. So there's a basic core where DNA is wrapped around here, but then there are these tails that come out. And these tails were thought to be curiosities until David Alice and others, starting about a dozen years ago, realized that these tails actually are little molecular signals. They can be chemically modified in many different ways. Some of them mentioned here. Acetylation, we know a lot. We use histone deacetylase inhibitors in the clinic. But there's histone methylation, phosphorylation, ubiquitination, all types of different modifications. These show the various residues that can be modified, and these show the various enzymes that can be modified. So you can basically determine if a gene is on or off by virtue of this histone decoration around a gene. We know this now from genome-wide sequencing studies where you can precipitate the chromatin using antibodies specific against different histone modification marks and sequence that DNA and then map it against the genome. And so here you have what's called, would be sort of a typical gene. And what would it look like if it's on? It would tend to have acetylation marks at the promoter. There's a nucleosome-free region right at the promoter where RNA polymerase will bind. There's acetylation 5' and 3' to that. And then down in the body of the gene, we have particularly this K36 trimethylation, which is actually a signal to prevent the polymerase from starting a second or third time in the middle of the gene, which would give you all kinds of short transcripts, aberrant proteins. That's a bad thing. So this is sort of just a lot of marking. And a lot of these marks are not just simply chemically changing the chromatin, but also serve as landing pads for some of those other chromatin modifying and factors, and allows kind of stabilize the gene in the on position. By contrast, inactive genes do not have this K histone acetylation. When I say K4, that means histone 3, lysine 4, methylation 2 or 3 means it's either 2 methylations or 3 methylations. And there's a lot of histone acetylation here. By contrast, an inactive gene will have histone 27 methylation. This precludes the acetylation, actually recruits a lot of negative factors that lead to the further histone deacetylation. And we get this K9 methylation in the gene body, and often you get a lot of DNA methylation, both of the gene body and the promoter, that helps permanently shut off such genes. Now, this histone code is, as I said, it's chemical modifications that might change some of the chemical state of the chromatin, but there's actually, it's dynamically written and erased. So they're writing enzymes, acetylases, methylases. There's erasing enzymes. These were recognized first about 2006 with histone deacetylases. And then about 5 years later, demethylases were discovered. Those were not even known to be known to occur. And all these represent potential therapeutic targets. And very importantly, there's these chemical modifications are read by the transcriptional modification machinery. So these bromo, these various domains here actually read and recognize these chemical marks and help reinforce gene activation or repression states. So you can imagine this is a very tightly balanced situation, and anything that might upset this might really, set the cell into a non-physiological state. And Dr. Varma several years ago asked one of his NCI provocative questions. He asked whether we can develop approaches to determine between a driver and passenger epigenetic event. So what would we say is an epigenetic event? Well, what I might mean by epigenetic event is some kind of non-heritable change in chromatin that's altering gene expression states to stimulate a pathogenic outcome, such as cancer. And what would be driving this? How do we know what's driving and what just simply is a side effect of just a gene expression state? How do we know what's actually driving it? So our answering framework has been to identify and enumerate mutations in the epigenetic machinery. So do the enzymes that actually create these marks, that write them, read them, erase them, do those get mutations? What do these proteins do normally, and what do they do abnormally in cancer? Now, this is the pan-cancer, this was done by the people at the Broads, called the pan-cancer gene mutation analysis. What they did is they asked what types of genes are most recurrently mutated in a given cancer and plotted against mutations that are recurrently mutated in any cancer. And they get this quadrant in the upper right-hand quadrant, which will blow up here. And this shows some very common mutations found over and over again in cancers, like P53, KRAS, P10, NRAS. You can see a bunch of those. These we knew about. But what was not really appreciated until the past 5 or 6 years is all these things in circle. These are all chromatin regulators. ARID1A is an enzyme that moves nucleosomes around. KDM6A, we'll talk about today, is a histone demethylase. There's DNMT3A, which we know is involved in leukemia and myelodysplasia. So over and over, we're getting these chromatin regulators popping up as amongst the most commonly mutated genes in human cancer. So when we take a look using online genomics databases, you can go to this one. This is the Sanger website called COSMIC, Compendium of Somatic Mutations in Cancer. You can go to myeloma, lymphoma, and you could ask what are the top genes mutated in those diseases, and we can ask are any of those gene regulators. When you look at lymphoma, and we've published this, we've talked about this in the past, EZH2 is mutated in lymphoma, and this is now being therapeutically targeted. We look at myeloma and just look at mutations. One of the things that comes up here is KDM6A. Some others come up as well, such as IDH1, which can indirectly affect chromatin. We see P53 curves a lot, but we'll talk about KDM6A and explain that. So KDM6A is a histone demethylase. It removes the K27 methyl mark, which I told you is inhibitory. It is found on the X chromosome. It stands for universally transcribed on the X chromosome. It is biallelically expressed. It's one of those genes that escapes X inactivation, so it's expressed from both the maternal and paternal X chromosome. And there is a Y chromosome component, homolog called UTY, which is very similar. Related is another protein called Jumanji D3. KDM6A is not only mutated in multiple myeloma, which we'll talk about, but many, many types of cancers, and it's just covered with mutations when you go into this database called cBioportal, which takes the entire cancer genome anatomy mutations and puts it into one easy-to-use compendium. This is like the only experiments I do is to go into this database and look for interesting mutations. This is very, very frequently lost in many cancers, most commonly in bladder cancer, but in myeloma in about 3 to 5% at diagnosis. Interestingly, though, almost all the myeloma, a good third or more of the myeloma cell lines that we use in the laboratory for studies have lost expression of this KDM6A. Additionally, our friend in the field, Leif Bergsagel, tells me every time he makes certain animal models of myeloma, by the time he can get them out to grow perhaps and transplant into another animal, they've lost KDM6A expression. So we think that this gene may, if not be the initial driver, at least a progression factor in multiple myeloma, this tumor of very mature B cells. So we decided to study what UTX might do in multiple myeloma. And just to remind you, UTX is part of a multi-protein complex. UTX complexes with a histone acetylase, P300, and a histone methyltransferase, KTM2C or D, to remove that K27 inhibitory mark and put on histone acetylation and K4 monomethylation, add enhancers in particular to turn on genes. So we predict that if you lose UTX, this whole complex may fall apart, and you'll have a failure of genes to activate. And those genes that fail to activate might be involved in tumor progression. So what we did from again, a friend of ours in the field, Jonathan Keats, we obtained a cell line pair called ARD and ARP. This was in the years not too long ago before you had gene editing to custom edit your favorite gene. These cells are paired from the same patient from different subclones of their myeloma, but one has UTX expression, the other doesn't. So we simply engineered it so we could put the UTX expression back, seen here, in a conditional manner. And we found that when we did that, these cells really slow down in growth, seen here, but it took a while. And we find this happens a lot with these gene regulatory proteins. It takes a week or so, or 10 days, for really the effects to kick in, perhaps because it takes a while for the chromatin to be modified and gene expression states to change. And what we found is there was a very, very modest decrease in S phase, increase in G1 in these cells, and no evidence of cell death. They just sort of slowed down in growth. There also were other alterations of the biology. We found that these ARD cells, D will stand for deleted for UTX, are stickier to fibronectin. They're stickier to plastic-coated surfaces, and the ARP are less sticky. If we add back the UTX, these cells become less sticky. Additionally, the ARD cells form more colonies in soft agar seen here, and if we add back UTX, we lose some of that colony formation. So increased adhesion, increased colony formation, all this sounds like a more biologically aggressive tumor. The UTX loss seems to correlate with a more aggressive tumor. And then we tested that by taking these cells that were conditionally can express KDM6A or UTX and add it back, and then measure the growth of animals implanted with these tumor cells in vivo using non-invasive imaging. And what we can see here is when we add doxycycline and we turn back on the UTX, these tumors grow much less well in animals, as seen here by the photon flux. And when we take these tumors and section them and stain them for proliferation, we see a much more noticeable decrease in S phase here. This is Ki67 stain, so these tumors are much less proliferative when they're put into mice and we turn back on the UTX. Also collectively suggesting that UTX is a tumor suppressor protein that's lost in multiple myeloma, as well as other tumors, may contribute to the progression and the biologically aggressive behavior of the disease. We then wanted to understand how does that happen and what genes might be affected. So we did RNA sequencing on the ARP cells, which are UTX replete, and ARD cells, which are UTX deficient, and then cells in which we added back UTX. And what we see is that there's a universe of genes, about 5,000 genes, 4,000 genes, that differ between these cells. And then about 1,300, 1,400 of these genes come back on and are turned back on when you turn back on UTX. And this is you could do statistical tests. This is like a p-value of virtually 0. This is not a random set of genes. Now, some of the genes that are lost when we lose ARD are genes such as E-cadherin. And when we turn back on UTX, we get back on E-cadherin. In fact, intriguingly, these ARD cells seem to have turned into sort of a mesenchyme. And this is an emerging concept in myeloma, as well as other blood malignancies, that as in solid tumors, where advanced tumors undergo an epithelial to mesenchymal transition, which increases their ability to invade the immune system, to migrate, to metastasize. Same thing might be happening in blood malignancies as well. And the loss of E-cadherin with UTX loss and its coming back on with UTX re-expression may be one hallmark of that. We found that genes that are downregulated upon UTX loss are highly involved in cell adhesion, in cell-cell signaling, extracellular matrix interaction, cadherin signaling pathways, and some of the genes that are lost, seen here, some of these such as versicams, another cadherin, these have been reported as mutated in multiple myeloma as well, suggesting that when you lose UTX, you might lose expression of a number of genes that may be involved in cell-cell adhesion and regulating the normal myeloma stromal, myeloma bone marrow environment interactions. And the loss of those genes may contribute to progression. Now, what we actually find is when we look at some specific genes, the UTX loss doesn't indeed alter chromatin marks. We see here when we have the ARD cells, we have an excess of this K27 methylation, which tends to decrease when we add back UTX, and we gain back that K27 acetylation and K4 monomethylation, suggesting that we are indeed, the UTX protein is indeed mediating the chromatin changes that we thought it would. What we also found by genome-wide ChIP-seq analyses, when we compare the ARD and ARP cells, what we actually found is that we didn't find a huge change. Here's minus UTX and plus UTX. UTX should reduce the K27 methylation. It does a little bit. These ChIP-seq tracks refer to a number of DNA sequence reads that are pulled down by the anti-Histone 27 antibody, and we can see there's a minor decrease here. But interestingly, on these genes, and there's some Wnt genes here, we see a major increase in Histone 27 acetylation. And this goes with old biologic data that shows that animals that are mutated for this UTX protein can actually be rescued from their phenotype by adding back a version of UTX that doesn't have demethylase activity. So it may not be only this demethylase activity, but the ability to scaffold and bring this histone acetylation complex to bear on a particular gene to activate the gene which might be most important. And we have new data suggesting a lot of the biological phenotypes we're seeing in these cells can be rescued in part by a demethylase dead version of UTX. So it may be this big multi-protein complex just fails to form and fails to turn on genes. We do have a failure of gene activation. But when you have a failure of gene activation, you will revert to a gene repression kind of phenotype, which is mediated by this complex called the polycomb complex and its central enzyme, EZH2. So our feeling is when we lose UTX, you might revert to this kind of more repressed complex. And if these genes are really important genes to stop tumor cell growth, we might be able to reactivate them by inhibiting EZH2. And there are EZH2 inhibitors in clinical trials. Some of them might soon get to approval. Maybe we can reverse this process by adding an EZH2 inhibitor and force some of these critical genes back on. So we did that with a GSK compound called GSK343, which we treat these ARD deleted or ARP replete cells with this compound. We remove the K27 mark seen here by immunoblot. And what we found, though, is only the ARD, the deficient cells, were killed and inhibited in growth by the EZH2 inhibitor. And this was associated with a cell cycle rest in the ARD cells, but not the ARP cells, and also with apoptosis. And this differs from the UTX add-back. UTX add-back just slows down the cells, but adding the EZH2 inhibitor slows down the cells and kills them. What we found is a panel of UTX deficient cell lines were selectively targeted by this GSK compound and not these two UTX replete cell lines. And we found those cells also underwent apoptosis seen here, and they also went cell cycle arrests. When we then we want to know, does this EZH2 inhibition, as we theorized, kind of copy giving back that UTX gene activator? So we do RNA-seq between cells treated with control compound or experimental compound, and we compare that with the UTX add-back, and we get a substantial overlap of those genes. So many of these genes that are turned back on by the EZH2 inhibitor are the same genes that we turn on when we add UTX back to these deficient cells, including E-cadherin, one of the genes I talked about earlier. So this epithelial mesenchymal transition may be potentially reversed by adding the EZH2 inhibitor. But we also found that there was a lot of genes turned on by UTX that did not overlap with genes turned on by the GSK compound. So some of these, the EZH2 inhibitor was turning on some additional genes in the the UTX deficient cells, and maybe those were behind the ability of these cells to die. With the EZH2 inhibitor, which doesn't happen when you add back UTX. And a lot of these genes seem to be involved in cell growth and killing, including the BH3-only protein BIC was turned on, and this Runx tumor suppressor protein was turned on over 100-fold in only the UTX deficient cells. So we did some kinetic studies to say, well, what's the rate of these genes being turned on over time? And we found that the cells were dying by about day 4. It's starting to die. Runx doesn't turn on until after about a week. BIC is turning on kind of modestly by day 4 or day 6. But what we saw that was really striking when we treated these cells with EZH2 inhibitors is they were turning off IRF4 and c-MYC, two essential proteins for myeloma growth. If there's one hallmark of multiple myeloma, it's a terminally differentiated B cell that continues to express MYC. And a normal plasma cell should not be expressing MYC. And so this is a hallmark and was shown to be a key driver of continued growth of myeloma cells, but this is getting shut off by day 4. And how's that happening? To figure out how this was happening, we took these cells and made them drug resistant by gradually increasing them in increasing concentrations of the GSK EZH2 inhibitor compound until we developed pools of cells that, that were resistant to drug. And we found that these pools of cells were still, the compound still works because there's no K27 methylation, so that inhibitor works. But the cells grow. They grow slowly, but they still grow, not as well as the cells that are untreated. And they're not undergoing any high rate of apoptosis. So these cells are in some kind of steady state. And what we found is that the GSK compound decreases IR4 and MYC, but when you release it from this compound, IRF and MYC comes back on. So something is repressing the expression of these key factors for myeloma. So we thought it would be some transcription factor that's aberrantly turned on that might then turn off IR4 and MYC. So we did some looking up in the literature, and we've had a number of candidates, and the candidate that really sticks out is a protein known as BCL6, which is a key transcription factor for the germinal center B cell. It's not really that involved in plasma cells and myeloma cells. But we found the GSK compound would acutely activate the expression of several B cell, germinal center cell specific transcription factors, BCL6, BATF, and IRF1. And when you remove the compound, that expression would then drift back to normal, seen here. And then the resistant cells, that upregulation had really drifted down back to basal as well. So acutely, you're turning on these transcription factors, and if you become resistant, you fail to turn on those transcription factors. Yes, Larry?
Have you ever looked to see in these cells, when you treat with the compound, to look for any other phenotypic differences associated with potentially the differentiation of cells?
Funny you should ask, and that was not a plant. So we went through our microarray data, and we went through and asked, well, what are the germinal center markers and what are plasma cell markers? And if you treat with GSK 343, you turn on all these germinal cell markers and you turn off all these plasma cell markers. And when the cells become resistant, you know, the plasma cell markers come back on. And when you remove this drug, the plasma cell markers come back on. So you're exactly right. What's happening is that these cells seem to be becoming more like germinal center B cells, not like plasma cells. And so that's exactly what's happening. We wanted to know, though, which of those transcription factors might be actually killing the myeloma cell. And to do that, we did this experiment where we force expressed them in combination with a green fluorescent protein. And so the idea is your cells are labeled green, and if your transcription factor, whatever you put in, is neutral to the cell, they'll just stay green because, you know, there's nothing's happening. But if you put in your favorite factor and make your cells green, but that factor acutely is tolerated, but over time kills off the cells, you'll get fewer green cells over time. So it's sort of a dropout assay. So we put in BCL6, we put in BATF, and we asked, do the cells stay green over time? And anyway, let me go through that. And what happened, we found, is BCL6 in particular, at 4 days post-infection with a virus having BCL6 and green protein, they're green, but by 5, 6, 7, 8 days, the green is rapidly going away. Those cells cannot proliferate anymore. The BCL6 is precluding their proliferation. So we think BCL6 is the key transcription factor. IRF1 was a little less consistent, but also had an effect in ARD cells. BATF, by contrast, the green cells stayed constant over time, so that wasn't it. But what we then did is we acutely affected these cells and looked at IRF4 levels. Sorry. Somehow the label didn't come off on here, but what this should have said is that this was IRF1 that did not, this was control, this was IRF1, I think that was BATF. It didn't affect IRF4 levels, but BCL6 did inhibit IRF4 levels. That's what that's supposed to say. So BCL6 seemed to be the key factor. So could this have clinical utility? In theory, it could. We took mice and we implanted on one side of them the ARP cells, which are replete for UTX. On the other side, ARD, the cells deficient for UTX. And we then treated with an orally available EZH2 inhibitor. And we found that those cells which were deficient in UTX, we had decreased growth as measured by photon flux. So we think that these UTX null tumor cells may be more sensitive to EZH2 inhibitors in vivo. There's been 2 recent papers suggesting that more broadly myeloma cells might be sensitive to EZH2 inhibitors, but they also use different inhibitors that have different potency. So far, we have some new data with gene-edited cells. It seems to be holding true that at least for these GSK compounds, the loss of UTX might differentially sensitize cells to EZH2 inhibitors. Though, though there may be other circumstances where other inhibitors might work as well in other cell lines. But at least for this system that we've studied here, we found that when we treat these cells with EZH2 inhibitors, we turn on these B cell, germinal center cell specific transcription factors, turn off IRF4 and MYC, sort of de-differentiate this plasma cell into more earlier B cell, and this doesn't seem to be compatible with B cell life, and these cells selectively die. So we think that, this was a lot of molecular biology, but the bottom line is I think EZH2 inhibitors could be a strategy in this form of multiple myeloma where you've lost UTX. This is, we think that these mutations are going to be showing up as the COMPASS study matures and we have more and more sequencing of advanced patients. Because the initial, and the other thing I'll also say is that this concept has been picked up by others. A recent paper in Science Translational Medicine showed that bladder tumor cells devoid of UTX are highly sensitive to EZH2 inhibitors. So this might be a more broadly applicable concept as well. Yeah.
Humans also downregulate IR4 and MYC as a mechanism by which they kill cells. What does that do to BCL6? I mean, how are the mechanisms similar versus different?
Well, the key thing is the downregulation of IR4 is a key factor. So in that case, you have Ikaros and Aielos that keep IRF4 on. And when you lose those factors, when you degrade them by image, you lose the IRF4. This case is, we looked at Ikaros and Aielos levels in these cells, and they didn't change with the EZH2 inhibitors. This is a different mechanism where instead of losing the activator, you're gaining an aberrant repressor. So it's just another way to get to it. Larry, did you have a follow-up?
Well, I was just going to ask you, because it looked like you're going to switch gears a little bit. So,
Yep.
The regulation of IR4 through BCL6, is that a direct effect, or do you think it's going to limp? Or?
It was previously shown to be able to bind the IR4 promoter. That was previous work. So we didn't go and do that in this study. But that's something we gained from literature. So we looked up literature, like what represses IR4, what's known, and these are the candidates that came up. So yeah, Larry's right. I am going to switch gears a little bit more about myeloma, but also some newer studies on another lymphoid malignancy. I got into this field and got to meet great people like Sager and got to know Larry better through the study of this protein that's eponymous for myeloma called MMSET and WHSC1, and now it's being sort of renamed as NSD2, just to make us all more confused. So this is a histone methyltransferase, which is very specific for one particular chromatin mark called histone 3 lysine 36. This gene is rearranged in about 15% of cases of multiple myeloma due to aberrant rearrangement to the immunoglobulin locus. And these 414 patients are amongst the poorer prognosis. I think they remain, they respond well to bortezomib, but they have not, they've been the people who've relapsed early from autologous stem cell transplant. And so this has been one marker of a more guarded prognosis in patients with multiple myeloma. We studied there's a number of cell lines available from patients who had this 414 myeloma. And we studied these several years ago and found that this MMSET protein is highly overexpressed in these cells. There's some different sizes because the immunoglobulin locus can put itself right here before the entire protein or cut off little bits of the N-terminus. But this part of the protein, including this important set domain, which is the enzymatic activity domain of the protein, are included in this translocation. You get this big upregulation of these proteins. And what we found in these cells is a big upregulation of this particular mark called histone 36 dimethylation. and a loss of the repressive mark K27. And this is a really striking difference that's not just at the antibody level, but validated by mass spectrometry. And what was known in the field is that there's sort of a race between these NSD enzymes and EZH2 to modify histone 27, which is repressive, or histone 36, which is actually an activating mark. This dimethylation is found in promoters. Trimethylation is found in gene bodies. This dimethylation is found near the start site transcription. Seems to activate gene expression. There's no such thing as a histone that's both K36 dimethylated and K27 trimethylated. We've looked for these by mass spectrometry. They just don't exist. So there's a dichotomous choice that a histone has to make when a cell replicates. Am I going to be on and have K36, or am I going to be off and have K27? And what we think happens is when you have so much MMSET in a cell, you're just overwhelming the cell. This is a very sticky protein. It's present high levels, and it crowds out the EZH2 and switches a lot of genes into the on position. And what we found by chromatin IP sequencing is that normally the K36 methylation dimethyl mark is found in peaks and valleys near start sites of genes. But when MMSET is high, this K36 mark is basically everywhere. It just sort of completely remodels the epigenome, and you don't have that normal structure. And one thing you find is that these peaks of K27 trimethylation found at repressed genes are blown away by the high MMSET levels. So we think that there's a gene activation by wiping out the EZH2 activity, so you get aberrant gene activation. And what kind of genes get activated? We published this, but I'll just say EMT genes, invasion, motility, metastases, DNA repair genes, things like that. So again, some kind of progression activity. More recently, we've studied MMSET NSD2 in a different context, and this came from actually came out about 5 years ago. So I was at an AACR meeting and was talking with Bill Sellers, who was then head of Novartis, who told me about this great resource called the Cell Line Encyclopedia, where they basically had done gene expression profiling, mutational analysis, copy number analysis on 1,000 different malignant cell lines. So you could take your favorite gene and ask, is your gene mutated in any of these cell lines? Many of us have been using the labs for years. So I did that and typed in, MMSET, and found there's a number of cell lines, 6 acute lymphocytic leukemia, 1 much beloved myeloma cell line called MM1S or MM1. I think Shamlin knows this cell line well, that have this a point mutation in the region of the DNA that encodes the histone methyltransferase domain. And so this was suspicious to us for being an activating mutation, something that might activate the enzyme. And what we did by molecular modeling with a molecular modeler is we found that this mutation is not found in the catalytic site where histone tail sticks in, but actually at the entrance site of the, where the enzyme starts to dock with a histone. And by molecular dynamic modeling, they actually suggested that this mutant enzyme could have higher affinity for histones and might have an altered enzymatic activity. When we take a look at the cell lines that have this point mutation, here's MM1S in myeloma, and here's these 3 ALL cell lines. They all have a high 36 methylation and a low K27 methylation, just like MMSET overexpression. But the mechanism is not overexpressing the protein. The protein itself has changed its function. Subsequently, this point mutation has been found in pediatric ALL, particularly it's enriched at the relapse of B-cell ALL in kids. It's also found in adult T-cell ALL and mantle cell lymphoma. In mantle cell, it's sometimes a subclonal mutation, meaning that 100% of the mantle cell lymphoma cells have a cyclin D rearrangement, and 10% of them have this mutation. And we think this mutation might harbor a progression, particularly given that there's one child reported in this Nature Communications report who had 1% of of the alleles mutated for MMSET at diagnosis and 100% at relapse. suggesting there's this enrichment after the therapy failed. So in order to understand how this protein might work, we use gene editing using CRISPR-Cas9, and we're able to selectively target that mutant allele and not the wild-type allele, able to cut that mutant allele and revert it back to normal. And when we did that and reverted that allele, we lose that K36 dimethylation and gain back the K27 methylation, proving that this mutant allele is actually causing, it is an epigenetic driver. It is changing the epigenome. It's changing this histone methylation by just a single base pair mutation. By mass spectrometry, we found that when 414 is in the wild-type proteins overexpressed, we get a 15-fold increase in the rate of unmethylated histone turning into mono, and about a 3-fold increase in the rate of mono into di. So that's when you overexpress. It's like Le Chatelier's principle from chemistry. You just have a lot of this enzyme is sort of pushing the reaction to completion. By contrast, the MMSEP point mutation, only the first step, which is 0 to monomethylation, is increased. The second step is at the same rate. And that's consistent with our model that this enzyme has now an altered affinity for chromatin. It's binding on, and it's having an altered enzymatic activity. And in vitro, we can actually show, get this protein made in vitro, and we do a turnover rate of the enzyme versus the amount of input nucleosome. This is wild type. This is the 1099 mutant. This is another mutant found in mantle cell lymphoma. Both of these can convert more histones to the methylation state per unit time than the wild type. So it's actually a hyperactive enzyme. This is very rare. There are only 2 cases of this happening in cancer. This and the EZH2 mutations in lymphoma. Every other kind of mutation in these types of enzymes is usually a loss of function. What we found by doing gene expression profiling by RNA-seq on these paired cell lines is that these are our non-targeted cell lines and disrupted cell lines. We have a big loss of gene expression when we remove this mutation, correlating with the loss of the K36 and the gain of the K27. So what we find is that gene expression is lost. And when we compare 3 different cell lines, we see a significant overlap in those genes that are lost when we lose the mutation, but no difference, no overlap with the genes that are gained. In other words, this small set of genes here that seem to be repressed in the presence of the mutation were probably secondary events and not important. These loss of gene expression here, or by contrast, gain of gene expression, if you think about when you gain this mutation, that may be driving the resistant malignancy. And what we found again is that those cells that have that mutation have this epithelial mesenchymal transition signature, yet again. And we found that biologically, these parental cells which have the mutation, I'm so sorry, are very migratory, they're sticky, and if we remove that mutation, they lose their migratory ability and they lose their ability to stick to surfaces and to stroma. So this point mutation is making these cells more migratory, more sticky, more invasive, we think, and that was, and it also stimulates their growth. These are the wild type, these are the mutant cells. We remove the mutations shown in blue, the cells grow less well. So all these things are revving up this growth of this leukemia cell. This is shown dramatically, I think, in a xenograft model. We inject tail veins of mice with these human ALL cell lines. These are the cell lines that have the mutation. The mice all have to be sacrificed within several weeks. These mice particularly aggressive because they have CNS symptoms and they have spinal cord compression. They need to be euthanized. The mice in which we remove the mutation live longer. And we actually measured the photon flux. This is the tumor burden. Seen in blue are the mice with the mutation. And they have to be sacrificed because they have symptoms here. These mice without the mutation can grow to very high body burdens of tumor without having symptoms. And one of the reasons for this is that these mice with the mutation accumulate cells in their CNS, in their brains. They get meningeal infiltration. And the mice without the mutation over here have more of a homogeneous pattern, expression a lot in the body, in the spleen, the bone marrow, and the brain, but it's not that concentration seen in the brain. So this is actually suggests to us that this might be a driver of dangerous relapse in pediatric ALL. Kids who relapse from therapy and often can relapse in their CNS. That's just simply, all right, I'm just going to skip over this. One other thing that's really fascinating about these cells is that these cells with the point mutation are resistant to glucocorticoid treatment. And here's one example here. These are parental cells and non-targeted cells, myosin plus dexamethasone. No killing whatsoever. But if we disrupt that point mutation, now these cells respond to dexamethasone. So we think that this point mutation drives invasion, motility, stickiness, CNS propensity to migrate, and glucocorticoid resistance. So all this would nominate, both in its myeloma and acute lymphocytic leukemia, if we could inhibit MMSET, if we could, we might be able to reverse this histone methylation switch, change gene expression. Could we inhibit tumor growth? And maybe we could sensitize to chemotherapy. Well, we modeled this in the myeloma model by using a doxycycline inducible knockdown of MMSET. And if we do this, if we knock down, if we leave the mice untreated, they all die, within a month. If we knock down MMSET with doxycycline, some of the mice have to be sacrificed, but we get some more survival. If we give these mice injected with these, in this case, myeloma cells with a knockdown of MMSET, we give them 1 dose of melphalan. Some of the mice have to be sacrificed by this time point. But if we give them doxycycline and melphalan, doxycycline to get rid of MMSET, melphalan as chemotherapy, we can cure mice. So if we had an MMSET inhibitor, I think it would sensitize 414 patients to chemotherapy, and it would also potentially sensitize children to perhaps dexamethasone therapy in ALL. In terms of doing this, we've been working for years. We have compounds that work in vitro against MMSET. They even will inhibit MMSET in a nucleosome substrate, full-length MMSET. These are not cell-permeable compounds. We can't get any effect in cells. And we're stuck, and the field is stuck, unfortunately. Now, we know MMSET is a good therapeutic target. We know it would do good in 414 patients, maybe ALL patients, but we don't have a drug for it yet, unfortunately. So to conclude, I'd like to say that I think I may have convinced you that there are epigenetic events that change gene expression, change histone modification, change widespread patterns of gene expression in cancer. These epigenetic events can result from genetic events, these genetic events being mutations, overexpression of histone modifying enzymes such as histone demethylases and histone methyltransferases. A lot of this balance is on the K27 methyl mark. The UTX affects K27 methylation. EZH2 does. MMSET indirectly does. That K27 methylation balance is critical. And we showed that the alterations in HMT activity can alter cell growth, adhesion motility, and drug resistance. We think if we had HMT inhibitors, as we showed in the case for EZH2 inhibitors in the UTX deficient cells, we may restore chromatin function and control growth. And with that, I think on time, I'd like to thank members of my previous lab at Northwestern and my new lab at the University of Florida, and our funding agencies and various other collaborators. I'd be happy to answer any questions. Thank you.